is something you're all dealing with so already in lab. You're using Clustal, and you're using, well, you will be using this week if you haven't gotten to it yet, a program called star, or star.perl.pl, which I wrote. And so I'm going to be explaining various ideas behind multiple sequence alignment, and in particular how they are finally realized in the star program. But star is kind of typical of many alignment methods that follow a certain principle. So first of all, what is multiple sequence alignment and why do we do it? Well, normal sequence alignment involves two strings. Multiple sequence alignment involves multiple strings. That's why they call it multiple. And the definition is really the same, or sort of the natural extension of what two-sequence alignment is. When you have several sequences or several strings, you're still permitted to put in spaces either before or in the interior, or after, or in the interior of each of these strings, so that the total length, after you put in spaces, of each string is the same. And that defines, therefore, a first column, a second column, and so on. And then you can talk about the characters that are in any particular column. So let me give you an example. Here's A, G, A space A, AT space AA, TG, CT, G space G space G. Okay, so we had three sequences here. They were of unequal length. Well, these two actually were equal length, and this one was one shorter. And we put, in this case, just a single space here. Here's a single space where we put in two spaces. And now we have well-defined columns, so we can talk about the characters that are in each column. And this is a multiple alignment. Now, several questions arise. One is, why do we do multiple alignments? Secondly is, how do we do multiple alignments? Third is, who's going to pay for it? No, that's lunch. Why do we do multiple alignments? How do we do multiple alignments? What do we learn from multiple alignments? I guess that's part of the why. And just one quote I remember from a well-known person in proteins, bioinformatics, who said, "Two sequence alignments whisper. Multiple alignments shout out loud." In other words, there are some relationships which you can see vaguely when you align two sequences. But when you have a whole set of sequences that come from a family, a set of proteins that do similar things or are related evolutionarily or somehow, and you can line them up in a certain way, there are certain patterns that just jump out at you. They're no longer lost in the noise of the rest of the sequence. They're just obvious. And some of them are the active sites of proteins. We talked about this at the very beginning of the quarter, that as sequences evolve, some portions of the sequence evolve more slowly. They're more conserved because they're more functional. And if you just take a look at two sequences, it's sometimes very difficult to see, because if the sequences haven't evolved too far from each other, then you'll see a lot of regions that are the same. You won't know whether they're the same just because the sequences haven't evolved very much, the time hasn't been very great since divergence, or because they're functional. But if you take multiple sequences, one from a rat, one from a pig, one from a mouse, human, and so on, and each of the non-functional portions are mutating somewhat randomly, they're going to mutate differently, and the functional portions are going to remain conserved. And then when you line them up, if you line them up appropriately, the conserved parts will just jump out at you. So there's a lot of things that you can learn from multiple alignments that you can't learn conveniently from two-sequence alignments. There are some other contrary reasons why it's not so good to try to do multiple alignments, but at any rate, multiple alignments today are really a central and critical part of how people do sequence analysis. Okay. So I guess the best motivation, the simplest motivation is that many important patterns or motifs, conserved portions, domains, whatever you want to call them, are faint. And so you really won't be able to see them when you just do two-sequence alignment. Or you won't be able to distinguish that those are a conserved important signal, a conserved important pattern. You won't be able to distinguish that from other things that are conserved just because the two sequences are not that highly diverged. All right, so here we have three-sequence multiple alignment. And in the notes that I will put up on the web, you'll see other ones as well. Now, in the case of two sequences, we had a very nice, well-defined objective function. We've been using similarity, which is to put in spaces so as to maximize the number of matches minus the number of mismatches minus the number of spaces. And when you found an alignment that maximized that objective function, that was an alignment which best tried to display the similarity between the two sequences. We, of course, also had substitution matrices which talked about whether this was a significant substitution or not, one that should be highly penalized or whether it's a very natural one. In the case of multiple alignment, there's not as nice an objective, single objective function that people have agreed upon, that people have found to be natural and informative. Okay, so actually in two-sequence alignment, there are multiple objectives as well. We've been using similarity, but there are others that people use as well. But similarity is very, very widely followed. In multiple alignment, there isn't as nice, or at least as agreed upon, objective function. No universal accepted objective function. Can anybody suggest one? Just imagine you're inventing multiple alignment. You turn back the clock 20 years or so. You're the first person to think that it might be a good idea to line up more than two sequences at a time. And you know about dynamic programming. You know how to do two sequences. What might you invent as an objective function, as a kind of a mathematical criteria for trying to get alignments of more than two sequences that tell you something interesting biologically? Hmm? Okay, the average of the pairs. Now, what do you mean by that exactly?
It's nice to compare 2 by 2. The average. It's not a good idea.
So you would do something like take two sequences at a time and do their alignment and find what their alignment value is and then average those values. But averaging those values wouldn't necessarily give you an alignment of all the sequences. So actually we're going to get to what you suggested, is that will play a part. But if I have an actual multiple alignment, like this one in front of us, what do I use to score it? What do I use to say, to try to determine is this a good one or not a good one? What do I use then, therefore, in an algorithm to try to induce it to find a good one? So what I'm really shooting for now is what's our measure of goodness when you have a multiple alignment? For two sequences was, the number of matches minus the number of mismatches. Yeah. Okay, the suggestion was to go to a two-letter alphabet, basically purines and pyrimidines, and see if they matched up. Okay, but still, if I looked at a particular multiple alignment, how do I actually turn that into an operational rule? What do I do? I think, actually, if I can try to get to the motivation of what you're looking at, if you have multiple sequences, then you're imagining you're sampling over a larger evolutionary time period, because if you think of the species where these different samples or different sequences are taken from, they're over a larger distance in an evolutionary tree than if you're just taking two of those sequences. And so you're trying to not be so concerned with the actual details of the individual characters, but some aspect of the sequence that might be preserved over longer history. And actually, people do that, particularly for proteins. They take the 20-letter alphabet and turn it into something that maybe only 6 characters, looking at things that are more preserved over time. But still, I want to know operationally how you look at a particular alignment and come out with a number so that high numbers are good and low numbers are bad, or the other way around. Anybody have an idea? Well, okay, to tell you, the first idea one of the first ideas that people came up with is still basically the idea that's with us, and not everybody's happy about it. But it's this. Since we have a criteria for evaluating the goodness of two sequences when they're aligned, and that criteria in its simplest form is the number of matches minus the number of mismatches minus the number of spaces. Since we have that criteria for two sequences, why don't we just take these, when we have a multiple alignment, we'll take them two at a time, okay, and we'll just say, what does this alignment look like as a two-sequence alignment? What's its value? What's this value as a two-sequence alignment? And then looking at these two, what does this look like as a two-sequence alignment? So as a two-sequence alignment, this one has one match, one mismatch, one space. Now we're at -1, back to 0, +1, -1, -2. If I did that right, -1? It should be, okay, -1. So these two sequences aligned in this way, but evaluated as if it's just a two-sequence alignment, should give a -1 if we did this right. Let's look here. This is one mismatch, so this is -1, 0, -1, -2, -3, -4, -2. I did that right? No? Never get these specifics right. All right. -3. And then these two, that's a -1, -2, -1, back to -2, -3, and -4. Now, in two-sequence alignment, we never put a space opposite a space. That's just silly because you just get rid of it. In multiple alignment, it certainly can happen that you have a space opposite a space. What you don't want to have is a whole column of spaces. So, we usually count these as zeros because they're, in a sense, identical. We don't want to count them as a +1 because there's nothing wonderful about having two spaces opposite each other, but it's not something you'd penalize either. So these are just usually ignored. Well, now that I have each pair evaluated this way, then I would sum up those -5, -8. So the goodness or the badness or whatever, the score for this particular multiple alignment is, in this case, -8. Okay? So this is what's called a sum of pairs. Objective. And it is basically given the multiple alignment of n sequences or n strings you consider it that multiple alignment basically as n choose 2 pairs, aligned pairs. Of strings. So when there's n strings, the number of pairs of strings is n choose 2. And then evaluate the induced alignment for each pair. And then sum those n choose 2 scores. Okay? So consider that multiple alignment as n choose 2, I should say probably induced aligned pairs of strings. So that I have this multiple alignment of 3 strings, but it induces this pair, this aligned pair, this aligned pair, and this aligned pair. Okay? So 3 induced aligned pairs. And with this viewpoint, what we want to do, if our pairwise objective function is to maximize the number of matches minus the number of mismatches, and so on, then what we want to do is find a multiple alignment which maximizes this sum of pairs score. Sum those n choose 2 scores, and the result is called a sum of pairs score. And then our problem, so this gives us the mathematical model. Okay, going back to the very first lecture where we have a biological issue we're trying to get at something that a multiple alignment will reveal. And then we have a mathematical model. Mathematical model is that we can evaluate the goodness of a particular multiple alignment by a sum of pairs score. And then we have the algorithmic problem. Find, whenever you're given sequences, find a multiple alignment that maximizes the sum of pair score. Okay. So I haven't told you how we're going to do this, but right now we just want to focus on the sum of pair score criteria. And going back again to the first lecture, whenever we have a particular mathematical model or criteria, even before talking about algorithms, you want to think about whether that criteria captures the interesting biology. And I know it's very hard to do off the top of your head, but in the sum of pair business, criteria, you want to at least try to think intuitively whether or not this is likely to reveal important aspects of biological aspects that come from sequence or not. And you probably have to play with a few of these things before you see the end result, or begin to have some real opinion. Now, I should mention that Clustal, which is a program that many people use for multiple alignment. You're using Clustal X, by the way. Has anybody heard of any other Clustals? Yeah, Clustal W is an earlier version. And my understanding is that Clustal X and W give you the same alignment, so you won't get a different alignment by using a different program. But X is more graphical. It gives you a more graphical display of the results. So if you're ever faced with Clustal W, you can at least be assured that you're going to get the same end result as Clustal X gives you. But Clustal X does not specifically or explicitly solve the problem of finding a multiple alignment that maximizes the sum of pairs score or sum of pairs criteria. And we'll get to later on what it does do and why it doesn't try to do this. There are some programs out there that explicitly do this. And one of them is called MSA, multiple sequence alignment. And I think you can find that at the Baylor website. Which one?
I think University of Santa Cruz.
University what?
UC Santa Cruz.
Oh, Santa Cruz may have it. Yeah, many places may have it. So look around for it if you want to try it out. Okay, so it explicitly follows the sum of pairs criteria. Now, the other thing I should mention about sum of pairs is that when we evaluate two strings, we've always done it in terms of maximizing objective function. It's also possible to have a minimization criteria for two strings, where what you're trying to do is minimize the number of spaces plus the number of mismatches. Okay? That's actually called the edit distance. And we haven't emphasized that in this class, but some people follow that approach. So if we're just trying to minimize the number of mismatches or the value of the mismatches, if you get them from a substitution matrix, plus the value or penalty of the spaces, then your objective function is a minimization objective function when you're looking at a pair. And when you are looking at a multiple sequence alignment and you use the two-sequence objective function for each pair, then all the way over here we'd want a multiple alignment that minimizes the sum of pairs scores. So whichever you're using for the two-sequence, whether it's a maximization objective function or a minimization objective function, it translates over here. And I'm mentioning this now because, in fact, STAR does use a minimization objective function for various theoretical reasons. But you could easily convert it into a max objective function. All right. So we have the what. We sort of have the why, although I'm not spending a lot of time on why multiple alignments are useful. Probably, many of you have seen lots of multiple alignments. How many people have, in their real biological work have seen multiple alignments, stared at them, tried to find something useful from them? Just 2 people? 3 people? 4? 5? Okay. Well, anyway, if you're at all into the real biological end of sequence analysis and dealing with sequences, you'll see a lot of multiple alignments. Very, very common. And in what we're going to do for most of the rest of the quarter, multiple alignments will be our basic object of interest. The two sequences are useful in database searches, and they provide the basis for multiple alignment, as you can see in here. But multiple sequences, working with multiple sequences at a time, is really critical. Okay, so now what I'm going to try to explain is at least one approach to this algorithmic problem, how to find a multiple alignment that either maximizes or minimizes the sum of scores, sum of pairs score. And again, I have notes that I'll put up on the web. And so there are 2 things I want to talk about here. One is an exact solution. Well, at least I should mention it. And I already have told you that MSA finds an exact solution. And then I'm going to talk about another method that is an approximation. It is not guaranteed to find the exact solution by an approach called progressive alignment. Okay. Can anybody guess or suggest how we would solve this thing exactly? We have this objective function. By exactly, I mean we really want to find a multiple alignment that maximizes or minimizes whatever it is your objective function is, the sum of pair score. Anybody know just at least a general technique?
It's like two-dimensional. We make a three-dimensional table and the three values, but it's more complicated.
Yeah. So if I said you had to explain this idea in 2 letters, what would those letters be? DP, dynamic programming. Okay? So dynamic programming, when we looked at 2 sequences at a time, we built a 2-dimensional table, and conceptually we were asking what's the best alignment you can have when you are allowed to use the first I characters of one string and the first J characters of another string. And we learned how you can incrementally go from a particular pair of values to one bigger one. Okay? Now we're going to do this. We can do essentially the same thing in 3 dimensions. We can say, I'm going to build a 3-dimensional table, 1 dimension for each one of the sequences, and I'll now have 3 variables, I, J, and K, say, and conceptually we'll want to find the best multiple alignment, according to our objective function, when we're permitted to use the first I characters of one string, the first J characters of another string, and the first K characters of the third string. And then we can write recurrences on how you can go how you can express that value for I, J, K in terms of values for smaller. For example, one possibility is that you line up the ith character with the jth character with the kth character. All right? So you would have, in the recurrences, Dijk equals the max or the min, whichever, over several possibilities. So I'm going to write those possibilities over here. Di minus 1, j minus 1, k minus 1, plus whatever you get for lining up the ith character from the first string with the jth character from the second with the kth character from the third. This is my notation here is a little bit undefined, but I hope everybody knows what I mean. So if I took the ith character from the first string, whatever it happens to be, and aligned it with the jth character in the second string and the kth character in the third string, okay, I would get some value from that. You'd see, are those equal or unequal? Are those equal or unequal? Are those equal or unequal? Okay, you would get some value for putting all those together. And then what's left over is the I minus first characters of the first string, the j minus first characters of the second, the k minus first characters of the third. Okay? And that's one possibility. What's another possibility? That there's some spaces, yeah. So that it would be like, well, I should draw a different picture. That it's going to be like this, a space jk. Okay? That's one possibility. So you would figure out, well, what's the value of that? Okay? And then what we have here is d, I. You're using all of I. Okay, all the I characters in the first sequence, but you're only using j minus 1 and k minus 1 of the second. And so on. I hope everybody's getting the idea. I think, if you get the idea here, and I gave you an hour, you could write out all the cases. Okay, there are more cases now than there were for 2 sequences, because now we have 3 for a single space. We also have 3 for 2 spaces. Okay, so 3 for a single space, 3 for 2 spaces, that's 6, plus this one, that's 7. So there's actually 7 cases you'd have to write out. And in the program that you would write, you'd have 3 loops because you're going to be varying the first position, second position, and third position, and so on. It would make a good homework exercise to ask you to actually do this, but we don't have time in class. And that's just for 3. Okay? If we had 4 strings, then you'd have something with 4 positions, and so on. And in the notes, I actually do have some pseudocode that lays out the dynamic program for 3. I don't remember whether I have ever written that into Perl or not. I'll look for it. But if I haven't, again, it would be a good exercise for somebody to try to do that. All right. Let me just ask the next question, though. For 3 strings, let's say they're each of length n, how many operations would it take to fill out such a 3-dimensional table? Well, the way to think about this is, again, for each combination ijk, the number of cases you have to look through is, in this case, 7. So you've got 7 lookups, you have some additions, you have some comparisons in the end. In the end, for each particular cell in this 3-dimensional table, you're going to do a fixed amount of work, fixed number of comparisons, fixed number of additions. It's more than there was for 2 sequences, but it's still fixed, independent of how big the 3 sequences are. Okay? So in the analysis, we next have to ask how many cells are there in this 3-dimensional table? It's the product of the lengths of the 3 strings. If I said they're all n, so now it's n cubed. Okay? But I can generalize this. So the number of operations for the exact dp solution the number of operations is proportional to the product of the lengths of the strings. This is actually, exactly true. This is true for the 2 strings. It's just we said it was the product of the lengths of the 2 strings. But now if we have 3 strings, it's the product of the lengths of the 3 strings. Because you have now this 3-dimensional cube. And if you have 4 strings, you have a 4-dimensional cube, and so on. And the number of cells is just the product of the lengths of the strings. Plus 1, actually, in each case, because you have the 0. And so, for let's say k strings of length m each, that means it's proportional to m to the k. When k is 2, m squared, we've seen that before. Now it's m cubed. For 4, it's m fourth, and so on. So anybody have any idea at what point, if we're talking about m being sort of the typical length of a protein, 300, 500 length, something like that, where does this become impractical? For what value of k? Which one? 3. Well, if you play with MSA, now MSA has lots of tricks to try to beat down the total number of operations that it does. But I think they tell you they can do about 7 or 8 to 9 protein sequences. And that's on a very fast computer as well. So the point is that although we know conceptually, we know in principle how to compute this thing exactly, there is a limit, a fairly small limit on how practical that approach is. And now for many purposes, if k is 8 or 9, that's fine. And sometimes you only have 3 or 4 sequences. And if you're happy with this objective function, you can use the dynamic programming approach to compute the exact value. And it's done within a practical time bound. But a lot of times people want to align 30, 40, 60 sequences. Some people talk about aligning thousands of sequences. Not sure why. But anyway, it's very easy to get into situations where k is much larger than what's practical for the exact approach. Okay? So just as we saw previously, you can do local alignment or global alignment between 2 strings very practically. But if you try to do that between a query string and everything in a database, that's not practical, so we have to go to some other system. In the same way, when you do multiple alignment, the exact approach through dynamic programming quickly becomes impractical. Even with some of the tricks, and I won't go into them, but MSA has some nice tricks in there to try to reduce the total number of operations it does. And it's able to, by using all those tricks and all that cleverness that people have thought about for several years, it's able to maybe to go from 7 strings to 12. Okay, but then it's really stuck. All right. So what do people do instead? You need something simpler. Well, when we went from dynamic programming to BLAST, we did something simpler based on these HSPs, these little, first of all, the k-mers that matched, and then spread out from there to the HSPs as opposed to the rigorous dynamic programming. And actually, some people do that in multiple alignment as well. They look for k-mers that are common to many of the strings, and then if they're roughly in the same positions, they would line those up. And if you have several of them, that defines what are called anchors or legs. People have lots of words for these things. And there's a whole several methods that are based on ideas like that. But I'm not going to talk about those. You might find them in the notes. A totally different idea is something called progressive alignment. And there are, again, many, many variants of this. Many variants. But they all have the same kind of flavor. They're very similar, which is that you have these multiple sequences. Let's say we have k of them. First of all, let's just align every pair. Okay? Find what the distance is or the similarity is between every pair of sequences. And that's not too expensive because that's just k-squared operations, k-squared alignments. K is, even when k is 100, that's a reasonably small number. Not like looking at it through a database, something you can afford. And in fact, if you, when you play with star, how many people have actually gotten to star yet for this week? Nobody. Okay. When you play with star, there will be an initial part of that computation where that's exactly what it's doing. It's taking every pair of sequences and aligning them pairwise with the same dynamic programming that we're all comfortable with at this point, pairwise alignment, getting out a score for each pair. Then, if it's maximization, that score, that paired score, which is the largest, has identified the 2 sequences that are the most similar. Okay? And if it's objective function, which is a minimization objective function, which actually is in star, the one with the smallest number are the most similar. Okay? So if you've done this, if you've done all pairs and you found the 2 sequences that are most similar, And your goal ultimately is a multiple alignment of K sequences. What should you do? And this is called progressive alignment. And your goal is to make something efficient. Anybody see a good idea? Yeah. Well, okay, now, progressive could mean character by character, but it means string by string. So progressive alignment will find the two strings that seem the most similar. And that alignment it'll fix. It's going to be part of the multiple alignment from that point on. So it just fixes that one. And then it tries to find what's the alignment distance between every one of the remaining strings and the two that have been aligned. Okay? And that's an easy problem in dynamic programming as well. Actually, I'm throwing in a bell and whistle here at this point. I want to keep it even simpler than that. Okay, so imagine next what it does is it takes the next pair whose alignment value is best, okay, and it fixes those. And then the next pair among the pairs whose alignment is best, and it fixes those. Now, at some point, the next best alignment pair, and this is from the initial computations that were done, the next best pair is going to be something that's already in one pair and something else in another pair, okay? And then you align those up according to their pairwise alignment. All right, I'm going to be much more detailed here in a minute. But the point is that you're going to take pairs of sequences at a time, use their pairwise alignment to build up a multiple alignment. And in the end, you'll have, of course, a multiple alignment. The definition of a multiple alignment is that you've inserted spaces. But your multiple alignment will be influenced as you go by good pairwise alignments only. You'll never look at 3 strings at a time or 4 strings at a time. So you'll only have computation times which is proportional to the product of 2 strings. You'll have many of those, but nothing that grows cubically. Okay, I'm sure this is too vague. So let me get into something very, very precise, which is actually the critical idea behind all progressive alignment methods. Okay. So something very precise. Definition. S is a set of strings of, let's say, K strings T is a tree. You may wonder what trees are doing in here. Well, this relates to biology, so we have to have something biological. Anyway, you'll see in a minute why. I mean a tree with like a graph. There's one. Okay, a tree type drawing. Points and lines, which form a tree that has no cycles. A graph with points and lines and no cycles. That's what a tree is. So S is a set of K strings. T is a tree with K points. These are also called nodes. Okay? And each node is labeled with one of the K strings from S. one of the k strings from S. So here's an example of one. There's a tree. It's got 1, 2, 3, 4, 5 nodes. So we expect 5 sequences, and each sequence is just a short thing. Okay, so far I haven't told you why we have trees or what good they do, but let me get another piece of chalk and I will. Okay, so if we have a set of k strings and we have a tree where the strings are laid out in this manner, then we say and then a multiple alignment of those S strings is consistent it's going to be consistent or really influenced by this T if for each pair of adjacent, and that's the key term, adjacent strings or neighbor strings in T, the induced alignment, pairwise alignment, is optimal. Okay. A lot of words. I have 30 seconds to illustrate this. I'm going to obviously have to redo this next time. But what I mean by this is I want a multiple alignment of these 5 sequences so that when I look at it pairwise, remember we're talking about sum of pairs, whenever I look at this particular pair, because these are neighbors in the tree, and these are neighbors in the tree, but these two, this one and this one, are not neighbors in the tree. But whenever I look in that multiple alignment at any pair that are neighbors, the induced alignment that I see is in fact the best 2-string alignment that's possible for those two. So let me just give you one. This is AX, X space Z, AX space Z, and this is in the notes, so you don't really have to take this down. A space X space Z, AY space Z, and AYX XZ. And if you look at a pair that are neighbors, AXZ, AXXZ, AXZ, AXXZ, and you think about what would be the best. Remember, space against a space gives you 0. What's the best pairwise alignment of these 2 strings? It's A against an A, 1X against an X, because you only have 1 and 1, but you have 2 in the other. So you're definitely going to have one space against a character, and then a Z against a Z. So this pair is as good as it gets for that pair. Even though it's in a multiple alignment, it's as good as it gets. If you take a pair that's not neighbors in here, AXZ, AY, XXZ, it's not guaranteed that it's the best, although it might be. Okay? I'll just leave you with the following question. Is it, in a multiple alignment, is it good to have pairs induced alignments of pairs that are as good as they would be if you were looking just at that pair alone. Is that a good thing? And secondly, is it easy to achieve that? Okay, well, before you come back next time, convince yourself that the answer to the first question, at least, is yes. We're trying to get the sum of pairs to be good, either big if that's their objective function or small if that's their objective function. If the induced pairs are as good as they would be just by themselves, you can't do any better than that.